Good morning, everyone. I'm going to talk about how to model brain regions from the single neuron, single synapses up to the full network. What we are going to do is to illustrate what has been our past history of implementing modeling, which are models which are very close to the experimental data. Maybe you are more familiar with as abstract models, artificial intelligence, and so on. This is a completely different way. This is a way in which we can actually model what we do in silico experiment, what we call in silico experiment. They are simulations which are very close which are very closely related to the real biological experiments. Our model of in which we focus most of our research is the hippocampus, and there are reasons for that. The reasons are that this region is directly involved in higher brain functions, learning, spatial navigation, object recognition, and so on. And it is implicated in a very widespread diseases like Alzheimer, epilepsy, and of course, cognitive aging and schizophrenia. Here you see a typical excitatory neuron, the so-called pyramidal neuron on one of the regions of the hippocampus called CA1. These neurons are very peculiar because their structure in terms of anatomy, but also electrophysiology, is exquisitely tailored to do higher brain function, as we will see in a few slides. So every time we think in terms of what a brain does during a cognitive function, we tend to think that the brain is involved as a whole. So basically, all neurons are working together to do some. But this is not so. All neurons cannot work together simply for energetic reasons, okay? So it has been found, especially in the hippocampus, that the coding on the information is very sparse. There are individual neurons that code for specific things. And I'm going to show you a couple of examples. One is this from a Nature paper in 2010. It's not from my lab, it's from the lab of Christoph Koch and Rodrigo Quiroga. This is showing, and I hope that you can hear the ticks that are representing neuron spikes close to the hippocampus. These are, let me explain you a little bit. These are recordings from humans with the electrode implanted close to the hippocampus because they were undergoing surgery for epileptic seizure treatment. And so this was a good opportunity to understand or get more information on how the brain implements cognitive function. In this case is the coding of individual pictures that the individual was presented during the recording. So, for example, in this case, this particular neuron was selected for Marilyn Monroe. So basically, what this picture with this movie is showing is that the hippocampus in particular code information in a very specific way. There are individual neurons that are coding for different objects or persons or events in such a way that when one knows that that neuron is firing, it means that something is happening. We can understand what the guy is thinking when we see some specific neuron firing, okay? Of course, this does not mean that that neuron is going to code for that specific object forever. It may be, the hippocampus is very plastic. So it means that after a few hours or after a few weeks, that very same neuron could code for many other things, that on a different object, for example. It could keep changing during its life. So since this is a cognitive function that is arising from a single neuron, we went a little bit deeper into the inner working of a different neuron. I'm sure that you know what a neuron is, what an action potential is. It's a signal that is generated by a neuron when its incoming signal is above a threshold. And so these neurons, specifically in the hippocampus, In addition to this very peculiar shape and morphology with different dendrites stemming out of the main trunk, they also have a specific distribution of ion channels in the dendrites. These are different from any other neuron in the brain in the sense that I cannot go into the details, of course, because I don't have the time, and maybe it is too technical for this audience. But if you look at different neurons, pyramidal neurons, they are more or less the same shape with a main apical trunk and several small dendrites coming out from the main trunk. And we look at the density of ion channels in the dendrites. We see that different neurons in different brain areas have a different distribution of channels. And the CA1 pyramidal neuron are the one with the most complex distribution of channels. And why is that? We can understand from this simulation here. Okay, so this is a typical simulation in which we are basically we have one of these neurons with two synapses, one in the distal part of the neuron and one in the proximal part. And we set up this simulation because we wanted to test what happens if we simulate a single neuron but embedded in a network. So, as you may know, in the CI, we say one pyramidal neuron, there are two main excitatory pathways. They are both coming from the entorhinal cortex, and they are related respectively to our memories or to what we are looking at that point. So basically, there are two incoming pathways that are coding for different things, but they are going to be combined, associated in such a way that this neuron can decide if it should release an action potential or not to the other brain regions. So they both start at the entorhinal cortex level, but then one path goes here in the distal part. The other part goes through the entire hippocampus, the dental gyrus, the CA3 regions, and then comes here in the proximal part of the neuron. So two signals in two different parts of the neurons, meaning the same thing, but coming from different sources. And they are coming at the same time, so they are synchronized. Okay, so let's see what happens in terms of an action potential generation. This is a simulation again. No, I was repeating the previous one. So if I use this neuron with the equipment of ion channels that are observed in a mitral cell, for example, which is another kind of pyramidal-like cell in the olfactory bulb, and the potassium channels there are uniform, expressed in the dendrites. So, and if we do different simulation in which we have two synapses with different timing, so this is a series of simulation in which we activated two synapses. One is activated here, the other one is activated progressively with different delays. And the full circle down here means that for this specific time difference, time delay between the two synapses, we get an action potential. So this series of simulation is telling me that when this neuron has the distribution of ion channels of the mitral cells, we have a very wide range of delays between the activation of these two synapses to get an action potential. So what happens if we use instead the ion channels and the distribution of ion channels that we see in a cortical neuron? Then this is what happens. Now the window for which you have an action potential is shorter. If we use the native distribution, the one that nature has decided to express in this type of neurons, you see that the window is even shorter. Okay, so this neuron is going to recognize two inputs only if they are delayed by these few milliseconds. Okay, so basically what you are seeing here is something in which this neuron is able to take care contribute to the synaptic integration of this neuron by using the action the ion channels expressed in the membrane to delay to deal with the synaptic input in such a way that the neuron is integrating the synaptic input with the proper time delay. Okay, keep in mind that the signal starts synchronized here, and you have two pathways, one in this direction, which reaches this region in a few milliseconds. The other part is going to go through two additional population of neurons. So it reaches here with a delay of about between 9 and 10 and 12 milliseconds. Okay, so if you don't integrate with the proper time delay, you are going to have cognitive problems because the signal here and the relation with the external world changes with the gamma rhythm every 25 milliseconds. So if you have a neuron which integrates signals with a larger time window, you are going to integrate information from one cycle of the gamma cycle of the gamma rhythm to the next one, and you are going to mix two different cognitive information. Okay, so this is why nature has decided, evolution has decided this distribution of channels should be in these neurons and not in other. But this is just an example of the powerful computational properties of this neuron. There are others, but I cannot go into the details. But how we can implement a model that is able to recognize an object. So we can use these little dendrites here. We call them oblique dendrites. And if we assume that each dendrite, by the way, these dendrites get almost 75% of all the excitatory inputs to this kind of neurons. We thought that if we assume that each feature in the input, it could be the eyes, the nose, the mouth, or even the properties of a different tower or different object, can activate different dendrites, then when the neuron is tuned to recognize this phase instead of this phase, then it can generate an action potential only when all these features are present at the same time. I can show you a simulation in which we are going to define the face by changing the eye, the nose, use its eyes, or the mouth. It's amazing how the expression changes by changing very little features in the face. So in this case, the mouth. Okay, save code, do the simulation. I've tuned this neuron to recognize this face, and we're going to see the simulation now. Okay, so basically there is going to be the performance path input is the distal here. And now after I think about 50 milliseconds, the three inputs corresponding to the phase that I've tuned on this neuron, this type of eyes, this type of nose, this type of mouth, they're going to be activating three different dendrites. Let's see. There it is. These three dendrites. The sum of these signals are going to generate a recognition signal in my neuron. Okay, so now let's just wait to be sure that it does, and I'm sure because I tested it before. So okay, so this is the recognition. So what happens if I change the mouth, for example? Instead of doing this, I can do that. Save code. Now only two features are going to be activated. Oh, okay. This is going slow because my laptop, unfortunately, is not too powerful. But still, this is running on my laptop. So it is a simulation in which we have practically about 10,000 nonlinear ordinary differential equation, because it is a full system with the object axle equations and everything. But still, it can run relatively easily. Okay, so you see that now only two dendrites are being activated, and so you will not get this phase recognized by this neuron. Okay, so let's move on. Another example of the very specific coding in the hippocampus is what is called the place cells. I don't know if you know what a place cell is, but if you record from a rat that is going around, from a mammalian that is going around close to the CA1 region and very close to the specific neurons, you see that that specific neuron is firing whenever the mouse is close or around a specific space in the environment. And if you record from different neurons, you see that they map completely the environment. So there are neurons coding for space and direction. Okay, different neuron coding for different locations in a different in the same space. So again, another suggestion that the hippocampus is doing coding very sparsely. So at the level of the single neurons. Okay. Again, a simulation in which we recreate the formation of a place cell, again, using dendrites to recognize different parts of an object. And we have a virtual environment here. And during the environment, the virtual mouse goes around. And then we give a reward when the mouse is looking, for example, this object. The reward is going to activate this distal input, and this distal input together associated with the clues that the mouse is looking at, in this case, the red bar, will form a place cell that after the reward is gone, is going to fire every time the mouse is around that location. And we can see here two examples in which, in this case is a movie, in which we have created two place cells, one for this bar and one for this bar. You see that now the place cells are formed coding for this part of the space or this part of the space. Okay, so now one example in which we can use single neurons to study and investigate Alzheimer, for example. So there are a lot of, I'm sure you know what Alzheimer is. You may not know that Alzheimer is touching in different ways the different ion channels that are expressed in a neuron. And there were several papers showing different activation or the different effects of Alzheimer in different ion channels. So we model all of them at the same time, and using a neuron in which we, let's see, one neuron, pyramidal neuron, in which we distributed a bunch of synapses, and we were modeling the progression of the disease by altering the membrane features of the dendritic tree more and more to model the Alzheimer progressively affecting the functioning of more and more dendrites. And what we, of course, we were able to model the disease in the sense that the neuron, so let me, this is control condition. We have some background activity, which occasionally will generate some kind of somatic activity. This is one spike. You see that you have a background activity in different dendrites. This is the depolarization along one path, one dendritic path from the basal dendrite through the soma to the tip of the neuron. Okay, so what I wanted to show you here is that we can simulate control conditions, but also Alzheimer. And when we apply Alzheimer, the neuron is not going to work anymore. But what we can do now that we have the full control of the system, we can also model some pharmacological application which is able to recover the normal conditions. And when we do this, I cannot show the entire simulation. When we do this, the neuron is going to go and generate spikes as under control condition. Of course, this is a big help for the pharma companies because they now have a way to go to the market with a pharmacological application in a much less expensive ways, because we can reduce by a lot the number of products, compounds that they are going to test during the search for a good treatment. Okay, so this is another example in the effect of electrical fields, but I don't have the time, so I need to skip this and go and building the network. So how we build the network? We go on and start from the experimental traces, and you see that there is a lot of variability in the experimental traces. We use almost 1,500 experimental traces to constrain our computational models of single neurons in such a way that we were able to be consistent with and reproduce the experimental variability that you see in vivo. We used to do this some tools that Luca is going to show you in practice, because we built a platform in which anyone can run this kind of modeling from an online application. And we, of course, we got also different morphology for different from UCL, from pyramidal cells, or from interneurons. And this is a typical example of experimental traces in the model in different cases for different type of neurons and interneurons. And once we have the model for different neurons, of course, we also need to, well, this is, I need to skip this. We also need to take care of the connectivity, because if you want to build a network, you need the single neuron, but you also need to model the synaptic integration, the synaptic interactions between different pairs. And so we also did that. And finally, we got to the full model of a CA1 hippocampal region in a rat. This is the platform that Luca is maybe going to show you later. And this is the full model. And now, instead of a single neuron, we have now full control and can do in silico experiments on the network. This is a typical simulation in which you see the activity, the spontaneous activity going on in the hippocampus, in the CA1, just as we mounted it. It began to be active in this way. And we can do any type of in silico experiment, including slices, electrophysiology, because we have full control of the model. The idea is that once we have the model, we want to connect it to a robot, of course, in such a way that we can do simulations of a robot having Alzheimer or epilepsy, and then test possible treatments again in silico. But we don't need the entire hippocampus to test for specific cognitive functions, because we already seen that hippocampus is coding using individual neurons. Okay, so instead of using the entire hippocampus, we this is another a few slides in which we now are reducing the full system because it is too big to run for real-time applications. We use using a nest and integrate and fire approach to model neurons in such a way that we can reduce the computational time by a factor of at least 3 order of magnitudes. In this case, we can still get very good agreement with the experimental data, but the simulation runs much faster. Of course, we are losing some information, but this is not the topic here. We want something that was implemented very fast. And this is a simulation of the mouse model now in NEST. There are 400,000 neurons, so it takes about 4 minutes using 2 nodes. So it is a relatively small system. But we also have been able to build a human CA1 model network, and I can show you the system right here, because the human hippocampus now has 5 million neurons, and it is not the shape of the mouse anymore, but it is much more complex with the typical convolution of the cortical regions in the of humans. And so we are now doing a simulation with this system to move to human. Of course, we're still needing the electrophysiological traces from human neurons. So we have implemented the model, but just using rat electrophysiological properties. It's a kind of chimera, but this is what it is. We are waiting for the data. And we also did robotic applications using the circuits that have been, I'm going to skip this. So what we did is to implement a very small circuit, implementing the basic circuitry of the hippocampus, the object cells recognition that recognize blue, red, and cyan, and direction cells recognizing the direction. We put together, using our intuition, a small circuit of about 40 neurons, and connected it to a virtual robot. And in this way, we were able, with, of course, synaptic plasticity, so the synapses are going to evolve during exploration. We connected to a robot going through a virtual maze. And so we found that in this way, this circuit, very small and very simple, is able to learn how to exit from a maze with a single train, with a single training set, okay? So it does not need, no matter how complex is the maze, no matter how many stops it has to do to recognize in the sequence to go out of the maze, with just one trial session, it is able to solve the problem. So I can show you the path here. So this is the training session. The robot goes in, it begins to memorize, learn the sequence of objects. It figures out that this is a dead end. It goes back, tries another path, in this case, this one. This is the good one because it has a green stop. But this ends the training phase. The next time the robot enters the maze, it goes directly to the right path. Okay, so just one training session. And this would give you the idea of the powerful circuitry that we use in our brain. And now, of course, we are going on with this circuit, with this idea, and we are using one of the Chinese version of the dog, and to try to add this circuit to a real environment, and see if the algorithm, or what we we should modify the circuit in such a way that the robot can go on, instead of a virtual environment, into a real place. Okay, so I think I'm going to stop here. Just a quick discussion, representation of my group. This kind of interdisciplinary work cannot be done by using a uniform background. I mean, I have a computer scientist, PhD in chemistry, PhD in mathematics, PhD in computer science and in mathematics, physics, biomedical engineer. Luca is here, he's not going to talk after that. And this lady here, Alessia, is my business manager. Basically, she is my boss, because I get the money. She tells me if, when, and how I can spend it. Okay. So, and with this, I thank you for your attention.